In today's lecture, we're going to discuss about water, especially its biochemical properties as well as role in biological systems as a medium as well as reactants and products too. Before we delve into water, let's quickly revisit the type of bonds. First one is the covalent bond, which ranges from 300 to 800 kilojoule per mole for their energy. And among those different covalent bonds, the carbon-carbon, carbon-hydrogen, or oxygen-hydrogen covalent bonds are the most relevant ones to the biochemical systems. And they range around 300 to 400 kilojoule per mole, which is about to be not too strong or not too weak. And there are several types of non-covalent bonds too, including ionic interactions, which is around 86 kilojoule per mole, van der Waals forces, and among the van der Waals forces, there are different types of interactions. Hydrogen bonds, which is 20 kilojoule per mole, dipole-dipole interaction, as well as London dispersion forces. So now let's talk about these interactions in more details. Most of weak forces are based on the charges of atoms or the molecules in the interaction. So let's take a look at an example here where there are two atoms in vicinity. Each atom, of course, has a proton as well as electrons. So naturally, there are going to be repulsive forces built up between the elements with the same charge, such as proton to proton or electron to electron. And also, there will be attractive forces between the element with a different charge. Now, as these two atoms get closer, the attractive forces will increase, meaning that these two molecules will pull each other with a stronger power. But when the distance is too close, now the repulsive force becomes significant. So they're going to push each other a little bit to keep an optimal distance for the interaction. And you can see the same phenomena here in this diagram, which shows the total potential energy. And the lowest point here is going to be the state where the two atoms have an optimal distance. And this is the state where there's a balance between attractive forces and repulsive forces. And all the molecules, including even neutral molecules such as chloromethane, has some charge separations. So when we look at these chloromethane molecules, the methyl group, this portion is partially positive, which can be written as delta plus, and the chloride portion is partially negative, so can be written as in delta minus. So it is basically now a little bit of a charged molecule. So this molecule will orient itself with respect to the neighboring chloromethane in a way that this positively charged region is located next to the negatively charged region. And if you have multiple molecules of the same kind, the chloromethane, then all of them will orient in those similar ways. Now let's talk about the different types of non-covalent interactions. The first type is an interaction between a permanent dipole and another permanent dipole. And interaction decreases as a function of distance pretty sharply as 1 over r to the third power, where r is the distance between these two molecules. So this interaction is the strongest non-covalent interactions among these different types. And the second one is an interaction between a permanent dipole and an induced dipole. And this induced dipole is not a polar molecule normally, but it has a transient charge, so it can interact with the permanent dipole. So it has a weaker interaction there. So its interaction decreases even faster than the first type as a function of distance with a power of 5. And the third type is the interaction between an induced dipoles. It's also called as London dispersion forces. And it has the weakest interaction there with the power of r to the 6th. So its interaction decreases extremely faster depending on the distance. And all these different types of interactions consist of van der Waals interactions, which ranges from 0.4 to 4 kilojoule per mole, which is kind of relatively weaker interaction compared to the covalent bonding. Another type of non-covalent interaction is hydrogen bond, which is a particularly important kind of interaction because of its critical role in mediating biochemical reactions. This hydrogen bond occurs between an electronegative atom bonded to a hydrogen, which forms the donor group, and also another electronegative atom with the lone electron pair. And this hydrogen is 20 kilojoule per mole, which is relatively higher energy for the interaction compared to the other non-covalent interactions. So this hydrogen bond can be represented as DH interacting with A, where DH is a donor group with the hydrogen atom, and A is for an acceptor. And the donor group is wickedly acidic, and the examples include the amino group or the hydroxyl group. And the acceptor is an electronegative atom with a lone electron pair. And the examples are the nitrogen atom or an oxygen atom. And this picture shows the two water molecules interacting to each other through the hydrogen bond right here. And this red-colored water molecule is a donor, and this blue one works as an acceptor. And the hydrogen atom here, which is attached to the oxygen, which is an electronegative atom, has the delta plus the electropositivity. And this oxygen atom that is on the other water molecule has electronegativity because of the lone electron pair on the oxygen atom here. So these two atoms interact to each other to form the hydrogen bond. And lastly, this hydrogen bond shares the features of both covalent and non-covalent interactions, which make it quite interesting and important for all the biochemical reactions due to this peculiar feature that has a relatively strong power for the interaction, but it also has some transient interaction properties. And the other type of non-covalent interaction is a hydrophobic effect, where the word hydrophobic means water hating. And this hydrophobic effect is a phenomenon where hydrophobes aggregate in water. And the hydrophobes are the molecules that cannot form the hydrogen bonds, and thus they have limited solubility in water. And these hydrophobes are the nonpolar and non-ionic substances, again, that cannot form the hydrogen bonds. And the examples are the hydrocarbons. But this hydrophobic effect is not a true force, but it's more like an entropic effect mediated by the tendency of water to exclude the nonpolar substances to basically minimize its exposure to the nonpolar molecules. And when the hydrophobes actually dissolve in water, the water molecules form the ice-like collaterate structures, basically cage the individual hydrophobic molecules. But this structure is highly ordered, so it's not energetically favored. So they will tend to move to the state with less potential energy. And less energy is required when these collaterate structures encage more than two hydrophobic molecules than just each alone. And thus, hydrophobes tend to cluster and form the aggregates in water. So why is water so important? Water is important because roughly 70% of the human body consists of water, and that percentage cannot drop below 55% to sustain life. And water determines the shapes of most of the complex biomolecules because of the hydrophobic effects that drives the globular or some other shapes of the proteins for their proper functions. And the water also works as a medium for all reactions in the cells. That means reactants and the products are transported by water. And the hydrogen cation as well as hydroxide are often themselves reactants as well as products too. And the reactivity of the functional groups such as amino group or the carboxylate is a function of this concentration of the hydrogen ion, which can be indicated as pH that we are going to discuss later on. And lastly, the water's reaction is particularly important and significant for the life of an Earth because of the water splitting event in the photosynthesis that leads to the carbon fixation. And this carbon fixation is basically the process that the inorganic carbons like carbon dioxide are converted into the organic form of the carbon complex. And this leads to the sugar production that is going to be the food for the heterotrophs. The heterotrophs are the living organisms that do not do the photosynthesis. And the byproduct of the photosynthesis, which is the oxygen molecules, are regenerated upon the reduction of O2 in respiration. Let's discuss more about the structure and the properties of the water molecule, which is definitely an unusual molecule. When we compare the water molecule to the structurally similar methane, or CH4, although this methane is a gas form at a standard state, they both have sp3 hybridization, which means that they form the tetrahedron too, although the angles are slightly different. So in methane, it has the 109.5 degree tetrahedral structure, but the water molecule, especially on HOH angle, is slightly smaller as 104.5 degree. Because of these two lone electron pairs pushes a little bit of these bonds. The difference is that first, the oxygen atom here is electronegative, so it draws the electrons from the hydrogens. And it has also two lone electron pairs, and that makes the water molecule polarized, and that leads to the 33% of ionic character to the this OH bonds. But the carbon in the methane is not electronegative, so it's not going to draw the electrons. And lastly, the water is dipole, as we discussed earlier, and participates in the hydrogen bonding, which is the key to the understanding of unique properties of these water molecules in the biochemical reactions. But the methane cannot form the hydrogen bonds, so it mostly stays inert. Here's the picture of water molecule. The oxygen atom makes the covalent bond with the hydrogen atoms right here, sharing the two electrons. And also this oxygen has the two lone electron pairs, which confers special properties of these water molecules. So let's quickly touch base on what we've learned so far. The hydrogen bond is a kind of electrostatic interaction between hydrogen that is bonded to an electronegative atom such as oxygen, and another electronegative atom can be oxygen or the nitrogen too. And this hydrogen works as a delta plus electropositivity to interact with the electronegativity of the other atom. And it has a small bond energy. When we compare to the van der Waals interactions, which is 0.4 to 4 kJ per mole, the hydrogen bond is relatively strong as about 20, but it's definitely smaller than covalent bonds. And this strength of hydrogen bond is very much dependent on the orientation of two molecules that are involved in the hydrogen bonding. As you can see here, the angle is definitely important. And the right angle that is aligned really nicely with the electronegative atom to the hydrogen with the other electronegative atom on the other molecule is the best way that the hydrogen bond is forming, have the strongest power. And the distance between these two molecules within the hydrogen bond is about 1.8 angstrom, which is longer than the covalent bonding. The examples 0.96 angstrom in the water molecules between the hydrogen and the oxygen atom, but it's definitely shorter than the van der Waals interactions, which is 2.6 angstrom. Again, hydrogen bond is relatively weak interaction, but it's extremely important because of its sheer number in the cell. Again, the 70% of the human body consists of water, which means that there are going to be a lot of hydrogen bonding that is formed and broken down. And in macromolecules such as proteins and DNA, there are hundreds and sometimes thousands per average proteins that you can see the hydrogen bonds that is formed. And also hydrogen bonds determine the shapes and the molecular properties of the macromolecules, again such as proteins and DNAs. Hydrogen bonding can explain some extended structure of water such as ice structure. Water can have up to 4 possible hydrogen bonds per molecule, and each molecule has 2 lone electron pairs as well as 2 hydrogen atoms, which work as a delta plus or delta minus, interacting with the other water molecules in vicinity. And in ice, all these 4 bondings are satisfied, so it's a highly ordered structure and also shows the tetrahedral patterns like this. And in ice, it shows the open form, and also it is much less dense than the liquid water because the volume is increased because of this highly ordered structure. And that's why you shouldn't put your beer in the freezer. In liquid water, only about 85% possible hydrogen bonds are satisfied, so it is less open structure than the ice structure. And the water molecules constantly rearrange with the high dynamics. So the hydrogen bond life cycle is only 10 picoseconds. And the hydrogen bond can be formed between 3 water molecules, 4, 5, or even 6. And since there are lots of hydrogen bonds within the water molecules, it needs energy to break. So the water molecule has relatively high boiling and melting points, which property is called as internal cohesiveness. You can appreciate this high boiling and melting points when you compare water molecule to the similarly structured methane. Water's properties make water one of the best solvents on Earth. Largely due to hydrogen bonding and its polar nature, the water has high electric constant, meaning that water can transmit electric force with little or no conduction or insulator. Water also can dissolve just about anything that is polar, just like amines, alcohols, aldehydes, or ketones that have carbonyl groups, as well as carboxylates. The solvent dissolves the solute to make a solution. So the solution is a homogeneous mixture of pure substances. And there should be no settlings in solution at all. And the particles that dissolve in the solution are molecules or the ions. Here are some relevant definitions. The solute is the stuff that is being dissolved. And the solvent is what the solute is dissolved in. And the colloid is a liquid with no settling just like a solution, but it has fairly big particles. So the true solution has a particle size smaller than 10 to the minus 7th centimeter, which is quite small. But the colloid have a particles size ranging from 10 to the minus 7th to the 10 to the minus 5th centimeters or so. And the suspension is a liquid, but it will have some settle downs of the particles over time, just like a dirt in water. And the particle size in suspensions are typically larger than 10 to the minus 5th centimeter or so. And another difference between solution, colloid, and suspensions is that when the light source is present, the solution will pass through the light completely, but the colloid will see the some fog that is highlighted by the light source, as well as suspension too. And for the solutions, you can put how much of solute that is dissolved in concentrations in many different ways, including percentage or volume to volume ratio, or the percentage weight to volume, or the weight to weight. And there's, of course, the molar concentration. And you should be able to convert between all these effortlessly. And you need to use the scientific notations almost all the times for the simpler calculations and the easier estimation of the numbers. So here are the quantities and units the chemists as well as biochemists use often. First one is Avogadro number, which is a mole with a unit of 1 mole, MOL, is 6.02 times 10 to the power of 23. And molar mass is basically the total weight or total mass of all these 1 mole molecules. So an example is a molar mass of water that is about 18 gram per mole. And one of widely used notation in chemistry as well as biochemistry is a molar concentration, which means the 1 mole, again, this total number of molecules per liter. And of course, we need to convert all the numbers to the scientific notations to simplify the calculations in chemistry and also biochemistry. And lastly, the atomic mass measurements are typically done in Dalton or kilodalton, and 1 Dalton is 1/12th mass of stable carbon isotope carbon-12. Solubility is the ability of a solvent to dissolve solutes. So it happens when the solute interaction with the solvent is stronger than with itself. And ease of dissolution or the solubility depends on the enthalpy changes delta H, entropy changes delta S, as well as Gibbs free energy changes delta G. And as you remember, depending on the enthalpy changes, the reaction can be exothermic, which releases the energy to the environment, or the endothermic that absorbs the energy from the environment. And entropy changes basically tell that this process increases or decreases in the degree of disorder. And using the combination of these two state functions, we can get the Gibbs free energy, which can tell and predict the spontaneity of the reaction. And many of you might think that this exothermic process is much more natural, but many solids dissolves endothermically too, because endothermic and exothermic reactions actually depend on these three factors. First one is the energy required to separate the solute particles from the crystal, which is a big particles, and that can be done by an ionic attraction between the solvent and solutes. And second is the energy that is required to separate the solvent particles from the bulk solvent to dissolve everything out into the solution. And the third one is the energy released in solvation process itself too. And the solubility is a balance between these all different factors and the energies. But again, going back to the question here, why endothermic dissolution, which is the sum of the energy 1 and 2, is larger than the third energy, still happens? That is because the disorderliness, which is delta S, the entropy changes, is greatly increases within the process and outweighs the delta H, which endothermicity. And again, the example is the ice pack, where there's a dissolution of urea that absorbs all the energies, but this reaction itself has a high entropy changes. So like we said earlier, the key to the understanding water as a solvent is about understanding the hydrogen bonding and how it works. So the water molecule can hydrobond with the suitable molecules, such as almost all the polar molecules. There are some examples. The first one is the water molecule interacting with the alcohol with the hydroxyl groups. And the second one is water interacting with the ketone with the carbonyl group. And third one is a carboxylic acid with a carboxylate that is interacting with the water for the hydrogen bonding. And the last one is an amine with an amino group that also making the hydrogen bonding with the water molecule. And rules for the hydrogen bonding is that the hydrogen atom in the donor group can have only one bond, as opposed to oxygen atom can have one or two bonds. And of course, the carbons, which does not have electronegativity, will not have any hydrogen bonding with water molecules. There are some more definitions to better understand the solubility, especially into water. The first one is hydrophilic or hydrophilicity as a noun, meaning that it's water-loving, and it's typically polar and ionic substances, so it's easily soluble in water and contains atoms other than carbon and hydrogen. So examples are the sugars, alcohols, and DNAs, and so on, such as glucose here. And the next one is hydrophobic, or the hydrophobicity. It means water hating, and it's basically insoluble in water and mostly constituted by the carbon and hydrogen and the hydrocarbons. The examples are hexane, mineral oil, etc., like a motor oil here too. And the last one here is amphiphilic, or the amphiphilicity, also known as amphipathic. Have both properties of both hydrophilic and hydrophobic. So sphingolipids is an example where one molecule has both hydrophilic part as well as hydrophobic part. Hydrophiles are the substances or the molecules that are hydrophilic, and they include polar substances and ionic substances. Polar substances have covalent bonds of atoms with different electronegativity, delta plus or delta minus, within the molecule or the substance, so they can work as hydrogen bond donors and acceptors. Ionic substances are held together by the Coulomb force or the electrostatic force, and this can be written as this in here, F equals Q1, Q2, which are the electric charges of the two substances that are in interaction, divided by the R square, where R is the distance between these two molecules. But in medium such as water, this Coulomb force can be written as this with the D electric constant included in denominator. And this D is important because when the dielectric constant is high, that means there's a weak Coulomb force that is set up between two molecules, especially ionic substances. So they're going to be easily dissolved in the media there. So D, the electric constant for the water, is pretty high as 78 because, of course, water is polar and the water molecule is dipole. So it actually shields the charge attraction between the sodium ion and the chloride ion in salt crystal. Again, make the Coulomb force between these two ions pretty weak and can easily dissolve into the water molecules. So the solvation actually is the process of making shells of water around these ions or the polar substances. And this process is pretty dynamic. But the D, the dielectric constant for methane, is quite low as 32.6, and the hexane is even lower, meaning that this electrostatic force or the Coulomb force will stay pretty strong and high. So it will be a bad solvent for dissolve these ions. Hydrophobes are the exact opposite of the hydrophiles. They are water haters, so they do not interact with water. And hydrophobes do not have charges, so they're not going to be any donors or acceptors for the hydrogen bonds. And the hydrophobes tend to exclude water and gather up in the presence of water, just like salad dressing. And the water forms cages, the clathrite structure around the hydrophobes, in an attempt to recover lost hydrogen bonds, like this. And thus, water becomes ordered around the hydrophobes, which is an unfavorable state. So it leads to the hydrophobic effect that we already discussed in an earlier lecture, that the droplets of the hydrophobes become round and fused together to minimize the surface to volume ratio of the water. And the globular proteins are folded under a same principle too, because the hydrophobes or the hydrophobic regions within the protein get inside and get together, and that whole process is driven by the entropy to lower any kinds of potential energies there. Amphiphiles have features of both hydrophiles and hydrophobes because they have hydrophilic heads as well as hydrophobic tails. Examples are soap and also sphingolipids, which are one of the constituents for the transmembrane. And since these amphiphiles have hydrophilic as well as hydrophobic regions, its mode of interaction with water is quite peculiar because it can exhibit both hydrophobic effect using tails and also the polar effect, which is hydrogen bond using the head group with the other water molecules. And interestingly, these amphiphiles can form higher order structures. When they make the blobs, the head group is facing outward towards the water molecules, and this structure is called micelle. And also, the amphiphiles can make the bilayer structures where there are two layers of amphiphiles that are ordered in a way that all the head groups are facing water molecules and all the tails are facing each other inside of this structure to hide from the water molecule. Solution has properties different from its pure solvent. Collectively, they are called colligative properties, which depend on the total number of solute particles in solution, but not the type of particles. And being a solution lowers vapor pressure according to the colligative properties and decreases freezing point and also raises the boiling point and also exhibits the osmotic pressure, where there's a solution inside of the column with a semi-permeable membrane, and that membrane is surrounded with pure water. In this case, the pure water will go through the semi-permeable membrane to lower the concentration of the solution. And you can put the stool bar there on top of the solution and push it down to the level as same as the pure water to measure the pressure here, which is going to be the osmotic pressure. And this osmotic pressure is proportional to the concentration of the solutes. And living cells must deal with this osmotic pressure because living cells have a lot of stuff, including ions and also biomacromolecules inside the cells. Thus, they have higher concentration compared to the environment, which will lead to the osmotic pressure buildup and the water goes through into the cell to burst them out. But the animal cells developed in a way that they keep it in the isotonic condition, which is basically the same as the environmental osmotic pressure. So it doesn't have to be burst out in that case. And for the plants and the bacteria, they built in a way that there's a rigid cell wall. So although the concentration is much higher within the cells, the cell walls blocking from the cell membrane burst out. Unlike osmosis, where solvents move into the solution, dialysis is about the diffusion of solutes. So a solution can be placed inside the dialysis membrane, and this membrane can be placed inside the solvent. In this case, the solutes from the solution will go through the dialysis membrane to diffuse out to the solvents to lower the concentration of the solution inside the dialysis membrane. And all these properties, including osmosis and the dialysis, are ingeniously used to purify certain substances or modify concentrations for any clinical or research purposes.